Hi, I'm Georgia Robbins Sadler. I'm the Associate Director for Community Outreach at the Moores UCSD Cancer Center in San Diego, California. Today I thought we would spend some time together talking about clinical trials. People don't really understand what clinical trials are. They're pretty much complicated to most people. So I thought we would just talk about them in a very comfortable fashion so that you would understand them as well as the clinicians with whom you might speak will understand them. So what is a clinical trial? Well, if you go to your doctor and your doctor says, take this pill, you take the pill and it works. How do you know it's going to work? What did we do to know that magically that pill would work? We did a clinical trial. Clinical trials tell us if new therapies are really effective, better than an existing therapy, as good as an existing therapy, or not nearly as good as what we already have. So clinical trials are very important to our understanding of how to advance healthcare, and especially cancer care. So what is a clinical trial? Well, let's imagine that you have a cancer and you would like to know what's the best treatment for it. You will be offered drugs that have been taken through various phases of the research process. The first phase, we look in the laboratory where there are just wet benches around, benches where people do research with petri dishes, little dishes that we put bacteria in or other parts of our body in. And from that phase, we have good discoveries. And we also have some bad ones, and we ignore the bad ones. We say, no point in exploring that further. But we look at the good ones, and we say, of these 10 good ones, this one is the most promising. And so we take that good outcome, and we move it to the next step, which is another set of studies. Same idea. Eventually, we get that product or methodology for treating a patient so well defined that we're ready to put it into an animal model. So animals get similar diseases to humans. And the discoveries that we develop for humans are first tested on animals who have the same or similar diseases. We take it through a whole series of tests. That drug might go through 2 or 3 or 100 different studies to find out if it works the way we think it will work. Once we discover that it works, then the next step is to take it to the human being. That's worrisome because what if it doesn't work the same way in the human being that it did in the animal model? So that's when we start to have clinical trials. The first thing that happens with a clinical trial is you get a very small group of patients who are willing to test this new drug. Now, who would that be? Well, often it's a patient who already has this disease, and there is nothing left to offer that patient but comfortable support. But there's really no good treatment option that we have left for that patient. And so that patient says, well, if there's not a good treatment option, maybe I'll try one of these experimental drugs because often they work. They've been through a lot of studies already, and we know they work in animals. There's a good chance they'll work in me, a human being. So that patient may very well volunteer to be our next study participant. Now, these first phases of studies, they often only take 5 or 6 patients. Very, very small studies. We're only concerned with 2 questions. Does the drug seem safe? And what's the best dose of the drug? So we start with a small dose. The next patient usually gets a larger dose, and the next patient a larger dose, until we reach the point where the patient begins to have side effects that the patient tells us, no more, that's enough. We shouldn't give this drug any more at this dose. We then take that drug dose back to the previous level because we know that was safe and it had the fewest side effects and it was very acceptable to the patient. So that's called a phase 1 study. Very, very small study. The next phase is a phase 2 study, and that's a little bit larger group. That may be 25 or 30 patients. And for that study, we're still interested in the question, is this drug safe for patients? That's always our first concern. But our second concern now is, does this drug make any difference to the patient's cancer? So if we give this drug to 25 or 30 patients, do we start to see a change, an improvement in the patient's cancer, or the side effects of the patient's cancer? The suffering of the patient's cancer may be reduced. We're looking for improvements. So assuming that we see good results in this study, that's the end of the phase 2, and we now move this new drug into a phase 3 study. And for the phase 3 study, this is a huge group of patients, very large. Sometimes hundreds, sometimes thousands of patients, because we're very concerned with finding even the slightest improvement that a drug can produce, because that may mean that 3 or 5 or 10 more patients will live with this new drug that would not have lived with the old drugs that we had available. So now we have a randomized controlled trial. The randomized controlled trial is called the gold standard. It's the best kind of research we can offer. It's usually done blindly. I'm going to explain a few concepts and then I'll show you a picture. In a randomized controlled trial, we want to be sure that the people who get the drug are just like the people who don't get the drug. So we want to be comparing two groups that really look alike at the beginning of the study. You've heard the phrase, we want to compare apples with apples, not apples with oranges. That's the same thing here. We want to be sure that at the beginning of the study, the patients in both groups are the same. Now, how do we get the patients into these two different groups? We do it with randomization. The computer randomizes for us and says, this patient will get this treatment, this patient will get that treatment. Now, if we know what the patient's getting, the patient can start to feel better just because their morale is better. They think, oh, I'm getting this exciting new drug, so I'm going to feel better because of that. The patient's doctor knows that the patient's getting a new drug. So the doctor starts to see the patient as feeling better. And the patient's family and friends, they all know the patient's getting this new drug. And they say, oh, this is wonderful, you look wonderful, you must feel better. And that often helps the patient to feel better. So we don't want to introduce confusion. We want both groups to think they've had the benefit of this new drug, because that way they both bring the value of optimism, hopefulness, a good attitude toward their illness. They're both feeling positive about the future. And that's very important, because you may have heard that the immune system helps to control our health and well-being. And to the degree to which we can help bolster the patient's immune system through supportive care, through confidence, through just feeling better, the better for the patient. So both groups start out at the beginning, and they both are given a pill or a solution that looks identical. No one knows who's getting the drug and who's getting either the standard of therapy or a sugar pill. Now, why would you get a sugar pill? Well, for the same reason we said earlier, we want both groups to be alike from the beginning, all through the study. The only difference is we want the patient in the experimental arm to get this new drug, but the patient in the other group will get standard therapy, and if no therapy is available, they will get a sugar pill or a sugar liquid that looks just like the therapy we're giving the patients in the experimental group. Now, the patients go through the study. So how do they get into each group? The computer randomizes them, and first it says, let's try and end up with the same number of patients at the end of the study in each group. And then it randomly assigns the patients. And then that determines which packet of medicine the patients get. So I'm going to show you a picture now that shows how that happens.